This next session is actually brought to us by Roche, and we have two incredible speakers who are going to be coming up and talking to us about gene transfer therapies, and so gene therapy and gene editing therapies. Do we have Dr. Gunnar? Oh, there you are. Okay, excellent. So with us we have Dr. Hernan Gonorazky, which we have all met over the weekend. He's the director of the neuromuscular program at the Hospital for SickKids. And I would also like to invite Dr. Zhenya Ivakine, a scientist in genetics and genome biology, working on applying genome engineering tools and strategies to therapy development for genetic conditions. And his lab is situated at the Hospital for SickKids. And he's going to be talking to us about gene editing. So thank you both for joining us.
Okay, thanks everybody. I know it's a Sunday and everybody's super tired. I understand that. Which one is this one? The big one, I assume. Okay, so I'm not going to talk about specifically for like the therapies are out there. I'm just going to tell you the basics so you guys can start understanding what's the difference between the different gene transfer therapy, or eventually just understand gene therapies in general, because it's actually quite a huge umbrella, and understanding like small of the topics around it, okay? So this is some of my disclosures for today related to this talk. So when we talk about gene therapy, actually there are a lot of things around it. You have actually antisense oligotherapy that I include here, okay? You can have gene transfer therapy, where you actually grab a gene and you try to put it back into the cell, and basically gene editing, where you actually try to modify the DNA itself. For my side of the talk, I'm going to talk more about gene transfer therapy, and Zhenya here will talk about gene editing. But this needs to be like clear that it's two different concepts. So the gene transfer, of course, has its pros and cons. Clearly, it's really small, and I don't even see it here, okay? But in terms of gene transfer therapy, you need to think about what's the condition and how it's actually working. For example, one of the first gene transfer therapies that was approved is for SMA, spinal muscular atrophy, and I don't know if you know it, but basically it's actually a motor neuron disorder. So the advantage there is that the neuron doesn't divide anymore, different than muscle. So basically you can actually put it there, and the transfer gene that you put back is not that it's going to be what we call incorporate into the DNA of the human body. So it's actually a piece of DNA that's going to be floating there. And if the cell divides, although the DNA divides, that piece of DNA doesn't duplicate, which eventually for dystrophies, like muscular dystrophies, that might generate a problem, not because the muscle actually regenerates. So that's actually a limitation different from, for example, the typical non-divided cells with the dividing cells gene therapy transfers. Okay, that's the first concept. Hopefully that was actually clear. If not, please just raise your hand and I'll take questions in the road, okay? So there are a lot of, there's more medications, but there has been quite a few EMA and FDA approved gene therapies for a lot of conditions. One of the most recent ones was actually Elevidys through Roche in US, although everything's on hold right now. But one of the things, and in terms of other gene therapy, I mean, it was the one from Pfizer, the one from Solid Bio, we have the RegenX, and it's like, okay, it's gene transfer in Duchenne. What's the difference between each of them? So what I want you to start understanding is like three main concepts that I know for Sunday afternoon is like a lot, but let's try to go through this so you guys start understanding, and you can ask the questions to your clinical team to say, what's the difference between each of them, and what's the most advantages for me? So the three concepts you need to understand is promoter, capsid design, and version of the gene. And we'll go for each of them. So the vector, okay, is basically the virus that we are going to use. And although like everybody's focusing about the tool that we are going to use, like it could be the gene or eventually CRISPR-Cas9, probably you have heard of it, is as important that the tool that you're going to use is how you actually get into the cell. And that's the vector component, okay? So you need to think the vector as in our case, it's a virus, but the vector can be different things. You can have a virus, or what we can have, for example, a nanoparticle, which is more a chemical component. One of the first patients with, which has, each of them has its advantages. And in terms of virus, you can actually have different viruses. You can have adenovirus, adeno-associated virus, is it AAV, sorry, lentivirus and retrovirus. And each of them has advantages and disadvantages. The lentivirus and retrovirus, a lentivirus, like a retrovirus, like HIV, okay? Don't be scared about it. We don't use it clinically, but at least in vivo, because you can have actually gene therapy in vivo, ex vivo. Ex vivo means you graft cells from the patient, you modify it in the lab, and you put it back, which is not applicable for patients with Duchenne. This is actually more for hematological, immunological disorders. That's actually used there. But the lentivirus, for example, will actually incorporate the gene into the DNA, which AAV adenovirus doesn't do that. And you say, okay, that's actually great. But the problem is that you can't use it in vivo because right now you don't know where into the DNA, that piece of DNA you are going to incorporate. And you actually might create a problem by producing another mutation in another gene causing a different disorder, okay? So we don't use that. So we focus in adenovirus and adeno-associated virus. The adenovirus actually is highly immunogenicity, like it produces actually a really big immune reaction. So you don't want to use that one. An adeno-associated virus has a really low immune reaction, so it means that your immune system that should protect you against virus or bacteria doesn't actually react so much to it. So the one we tend to use, actually the one we use is adeno-associated virus, AAV, and it has a specific capacity. So there's an amount of DNA material that you actually can put there, which is actually low compared with the other vectors. So AAV has a low toxicity, rare integration into the host DNA, so it means that doesn't actually go into the DNA and actually produce another disease, okay? And the ability to persist for months of years. So the vectors, in this case we are talking about AAV, is what it has different tissue tropism. What does it mean? Is that one virus can actually go into specific tissues in a different way, okay? So it might go that it will target more the muscle than it might target more than the brain, for example, okay? And and what do you actually want? You actually want to target as much muscle as possible and little of the other organs. Because in the case of Duchenne, no? Like that's clear. Why? Because if you do that, the dose that you need to give is actually lower. And if you give a lower dose, then the immune response is going to be lower. Is that clear? Perfect. Okay. So you have different types of AAV, actually. So the same way that you, COVID-19 is 19 because it's a type of COVID, but you can actually have different COVIDs, okay? Different what we call serotype of the virus. It's the same thing for the AAV. So you have different forms of AAV. You have AAV9, AAV8, AAV2. And most of the therapies are based for muscle therapy are based in AAV9 and AAV8 because they tend to actually infect the skeletal muscle and the cardiac muscle way easier than the other ones, okay? Now, you can actually modify that tropism to actually improve it even more, so it targets the muscle even more. And that's what we call virus with a high muscle tropism. And there are actually different ones, MyoAV, AAV-Myo, they're actually being developed. And the advantage of that is basically you just reduce the dose, and eventually the risk of adverse reactions are going to be lower. So this is actually like really busy, but I just want to show you. So you have these are two types of mice, okay? The white one and the black one. And then you have the typical, in black circle is the AAV9, which is the typical normal, no touching, no modification of the AAV vector. And then you have actually in the other colors, green, red, and blue, modified versions of that same vector. And then you actually see how good it's actually infecting different tissues. For example, the tibialis anterior, which is a muscle right here in the leg, okay? Or the diaphragm, which is the muscle, respiratory muscle. And you can see that the ones that actually have a higher muscle tropism, for example, the red and the blue ones, are actually really good compared with the other two. The same thing for the heart, and eventually the same thing for the liver. But although, and the other thing I want to show you, and although you have two different mice, although both of them are mice, they are from different strands, and they're actually different in how good is the vector to actually go there. That's why the thing that we still don't understand is the variability in human beings. It's not that it's going to be exactly the same in each of the person, how it's going to, the virus is going to be affecting the person, okay? So hopefully that was clear. So now we go to the promoter. The promoter, you need to understand, is like the engine, okay? So you want the engine and saying how much of that DNA is going to be expressed in the muscle. And it's not only that, because as the same as the engine, you can have like diesel or you can have like normal gas. The promoter can be specific for the tissue that you want that gene to be expressed. And why is that important? Because you don't want dystrophin to be expressed in the liver. You don't want dystrophin to be expressed in, I don't know, in another tissue that of your interest. Why? Because related to the expression of the protein itself, and we'll see that later, if you have an immune reaction, basically your defenses start attacking that protein that doesn't recognize, you don't want to be attacking, for example, the liver, because it's actually expressed in there. Eventually, you just reduce the tissues that the gene is going to be expressed. And then how much is going to be expressed? And you might say, oh, we should express as much as possible. Be careful, because the same way that the genes, when they are actually really low expressed, cause a disease, sometimes you have a really high expression, you also have a problem. So you need to know how much you regulate. So one thing you need to ask is like, is the promoter that is being used, is tissue specific? That's a question you might ask to if you are thinking going to a clinical trial. If it's expressed enough to actually reduce the viral load, and eventually how good is going to be the expression of the tissues, okay? So that's in terms of promoter. The last part is like which version of the gene. So like every gene, and this is evolution, has changes in the DNA, and that's why you actually have evolution. Like not every gene will actually work in the same way. So one thing in doctors we think is like in a genetic disorder is like healthy, non-healthy. And it's not like that. You need to start thinking the genes as actually, I don't know, like a gray scale. Some genes actually work better than the others, okay? So which version of the gene you actually want. And that's actually if you are putting the whole gene inside. The problem in the dystrophin gene is that it's a huge gene. It's just huge, okay? And actually, in AAV that has a capacity of around 4.7 kilobases, don't worry about the kilobases part, what I can tell you is just doesn't fit. It just doesn't fit the whole gene into the vector, okay? So what you need to select is which version of the gene you are going to put in. And that's actually a key. So by studies, by actually seeing patients and see how they are doing in terms of Becker dystrophin. Yesterday we were saying that actually the disease is not like just two. It's not Becker Duchenne. It's actually also like you have severe Beckers and you also have really mild Duchennes. Okay, by looking how the patients actually did over time, we know that in specific areas like of the protein, you can actually lose up to 15% of the gene and still be a milder form. Okay, so it's just by understanding how the patients actually did. So when we call microdystrophin, it's basically a smaller version of the gene. And this is actually are some of the trials. Pfizer, Sarepta, Geneton in the second place, then Solid Bio, Reginex. So I'm not going to go into deep into all this. If you guys want to ask me later, I'm happy to talk about it. Eventually, there's representative of people here from different companies. You can ask them as well. But you can see that NT is like sort of in the same, and CR is the same. but you have the rod domain actually is different between them. And then you have these blue lines with a zigzag component, and you can see that in some versions you have actually 3, some of them are only 2, some of them actually have a blue and a red one. That's actually how it's actually connected. I'm not going to go deep into that because it doesn't make sense to explain right now, but it's different versions of the gene. And why we're doing that is because we still don't know which forms of microdystrophin is going to work. Because expression of the gene is not enough. It might have a fantastic expression of the tissue, basically solving how we actually deliver, but it might be that the version of the protein that we're putting in, it might not do the work that we want them to do. Okay? Oops. So for that part for you to understand, which is the immune reaction. Immune reaction, again, is basically your defenses attacking like a foreign, in this case, the first part, it could be the foreign virus, okay? But you can also have a reaction against the foreign protein. In Duchenne, like in general, unless you have, in general, it's not always that, but you might have basically non-expression at all of the dystrophin gene. At all. Like if you do a muscle biopsy, there's no expression at all, okay? In some cases, like the protein, the human body will actually grab that protein and say, hey, like the one that I'm putting back, and say, what is this? I don't recognize that. And then it will start actually attacking the cells that is actually expressing that, okay? That's what we call an immune reaction to a foreign protein. And you need to think about this as the human body. Actually, you create tolerance over time. It's like the peanut butter. Like in Canada, I come from Argentina. We are not allergic there because I guess we get exposed to everything from the beginning. But now like here in Canada, you actually get exposed really early to peanut butter or like to peanuts. So to actually create that tolerance to peanuts, okay? We can try to get there with a foreign protein, but eventually how we actually do it is like something we're still trying to understand through clinical trials. And you can have actually quite a few immune reactions to eventually or the viral capsid, so basically the virus, or eventually the protein. And the time is actually quite different. Like usually against the virus, it's going to be really early in the first 7 to 10 days. And then against the protein might be actually little later when the protein is actually getting expressed between 3 to 4 weeks after, and even more after that, okay? And you can have, these are a typical immune reaction component. That's why wherever you're actually going to be, one of the things we actually did is we actually put a group of people together to actually understand this. You need to understand gene transfer therapy as an organ transplant, but just instead of doing massive amount of DNA, because that's what the organ transplant does, you can actually, what we call, have a guest versus host rejection, like when you actually have a rejection against the organ, you can have a rejection against the protein. So it's sort of the same way, but in a really smaller or scale, okay? That's why it's important whatever you're actually doing is like, hey, like what's the expertise that you have if my kid actually have a complication? Like, do you have everything set up? And just to cover everything. And across Canada, we need to build that expertise. Like we need to, thank you to you guys, like being exposed to this and understand all these complications, we actually build that, so that expertise to actually provide better care. So just with that, again, your courage drives discovery. Every step you take in this journey brings science closer to a cure. I mean, it's like without you guys, we can't actually do anything. It's basically, it's you guys that actually drive this. Like that's actually important. And like being so courageous to actually go into clinical trial. Like when you actually go there and I explain all these adverse effects that are so scary, and you still go through, it's like quite courageous from all of you. So it's a round of applause for you guys. Like that's I can tell you for sure. And with that, I just want to thank my team and the SickKids team, and that's it. Thank you so much. Now we go to the second speaker, and then we'll do questions. Thank you. Zhenya.
Good afternoon, everybody. So my name is Ivakine. I am a senior scientist at SickKids Hospital. And Lucy helped me to make this presentation. She is not here today, but I wanted to thank you as well. So what do we actually do? So my lab is working in a space of rare diseases, and we are interested to understand what genetic variants cause disease. So we are trying to identify those variants, and when we do, we are generating cellular models of disease. So we can actually put this variant in the cell, and then we're asking the questions, what is going to be the best way to actually rescue that condition? And more often than not, we are using genome editing strategies, and I'm going to talk about it. So we are trying to find a way, how can we correct that mutation, or potentially find a way to compensate for that particular mutation. More often than not, we generate animal models of disease, and usually it's a mouse model, so we are applying whatever we've learned in our disease modeling in mice, and hoping that we are able to rescue disease in mice. And then we want to bring this understanding back and actually try to apply the therapy in patients. So my presentation hopefully is going to be less scientific. I was trying to make it more entertaining. I will ask you afterwards if it worked or not. So if you think about our human body, every single cell in your body contains about 3 billion letters of DNA. So it's a stretch of letters, and there are only 4 letters, A, T, G, and C. And from those 4 letters, we generate diversity. Look around the room. You're all different. You're all similar, but you're all different. And there is a variation in human genome, and this variation leads to diversity. Some of you have brown hair, some of you have blonde hair. Some have curly hair, some have straight hair. People like me who are lucky have no hair. But it's all about DNA, and most of the time, this changes in DNA are completely benign. So they generate who we are. But unfortunately, in some conditions like Duchenne, small changes in DNA can lead to a disease like Duchenne muscular dystrophy. So if you think about DNA, the best analogy you can think about is a cookbook. So that is a template, everything is written in a DNA. And then when you're talking about RNA, that's the recipe. So DNA, it's instructions. Instructions are written and they are stable. RNA is produced from that instructions. It's a recipe, and from that recipe you can make proteins. You can make a cupcake, and we are going to call it dystrophin. And if you want to have a tea party, we can bake multiple different things, and then everybody is going to get something what they want. And because there are different cells in a body, we generate different proteins. So kidney cell is going to be different from muscle cell. It's going to be different from heart cell. So, and as you can see here, we can actually think about if something happened on the level of a cookbook, on the level of DNA, if we can actually fix something on that level, we are going to fix all the consequences downstream. RNA, like if you probably heard yesterday about antisense oligonucleotides, we are correcting on the level of a recipe. But it means that if recipe is lost, we had to print a new one, and we need to correct a new one, and so on. So that is the main difference. Not going to talk about Duchenne. You guys know the topic very well, but in general, so as I said, DNA mutation, they lead to inflammation, so it's a secondary effect. And what we are trying to do, we are trying to both work at the level of DNA, at the level of correction, and also at the level of inflammation, and how we can combine both of those treatments. And that basically, I already said it, so you don't need to watch it. So what is CRISPR, or genome editing? So genome editing is a tool that allows us to make a targeted change at the level of DNA. So we go to the root of the problem. If an individual have a mutation in the DMD gene, we are trying to correct mutation. So we are trying to fix the cookbook. So all the recipes that produce from that cookbook are going to become normal. And we hope that that strategy is going to ameliorate the disease. Classical genome editing system right now that have been used since 2013, it's called CRISPR. How many people know what CRISPR is? Raise hands. Amazing. Sometimes I ask and like, oh my God, we have to go all the way back to high school. So there are two systems, two different parts in CRISPR. There's a Cas9 and there's a guide. And essentially it's a two-component enzyme that is going to make a break in the DNA, and the cellular machinery is going to find a way to fix it. Cas9 is identical for most cases. GPS, guide RNA, is different. So we can target guide RNA to find a location in the DMD gene that is affected, and Cas9 is going to generate a break or some edit, and we are going to fix it. So that's the difference. So, and because there are different mutations, we can use different approaches. So here, for example, it's a duplication. In this case, we have two exon 2. So it's a duplication of exon 2. And you can read along. So that's actually going to be duplication 44. So here you can have some instructions. So until that, in a normal health, in a healthy individual, we can say the fat cat ate the rat. Poor rat. So what's happening if there's a duplication? We have AT is now duplicated twice. And basically everything downstream is scrambled. We are not producing normal protein anymore. So what duplication removal with CRISPR allows us to do is to actually excise that AT from the genome, and we can get again a normal message. So that's correction on the DNA level. Another example, some mutations in the DMD gene are single base pair changes, and we can use a new technology like base editing that is a little bit more precise than classical nuclease strategy, and to correct a single nucleotide mutation. For example, again, we have the same example, the fat cat ate the rat. Mutation, we can see change from T to Z, and base editing is going to correct that Z back to T, and we can get a normal DNA again. So that's what's happening with genome editing approaches. And we can use the same strategy about exon skipping. Not exon skipping on the level of a message, not antisense oligonucleotides, but we can actually remove an exon and to restore the normal message. So in this case, we are losing some of the information, but at the end of the day, we are not, the message overall still maintained, and we are able to make functional protein or functional chapter of the book from that. So we are working on, we are generating humanized models of mice. Basically, we generated a model that we put human DMD gene in a mouse, and we can generate right now targeted changes in this human DMD gene. And basically what it means that if we develop in therapy in cells, we can actually apply it to our humanized models very fast. So it's one-to-one. And not going to go through those details, but the first part is always is to find the best guide RNA, find the best GPS that allows us to do targeted modification in the genome. And essentially, here is an example. If you're looking at that dark band on the top, that's a restoration of dystrophin protein. So that individual did not have dystrophin protein being produced. But by using CRISPR technology and by targeting the duplication, we are able to remove the duplication, and we are going to be able to actually restore expression of fully functional dystrophin protein. So here is probably our best example about how base editing works. So on the, I guess, left for you, that's a mouse model with a normal production of dystrophin in heart. In the middle, it's a mouse model where dystrophin is not being produced, so there's nothing green you're going to see. And on the right, you are seeing that after base editing approach, we are getting about over 90% cardiomyocytes right now are dystrophin positive. And that is the power of CRISPR technology. So essentially, think about it. We took a mouse that was completely dystrophin negative, and right now almost every cell in the heart produces fully functional dystrophin. So that's what's becoming possible. And we are still doing a lot of optimization. So Hernan was talking about promoters. We are still optimizing promoters. So we want not just expressed in heart, but in all different skeletal muscles, because some of them are slow twitch, some of them are fast twitch muscles. We want to target all of them efficiently. We are modifying our capsid. We are making them myotropic, so better delivery to muscle, detargeting from liver. We are optimizing how DNA in the AAV is going to be, because we want production of full-length capsid protein, full-length capsids, eliminate empty capsids. We are trying to modify our genome editing component so they are less immunogenic. There is still work to be done. And finally, we are trying, we know that most of the Duchenne boys are going to be on anti-inflammatory. So we are trying to see how our genome editing is going to work in that context. So is it going to make genome editing even better, or we have to actually adjust something? So it's easy for me to talk about it. I'm not doing all the work. People in the lab are doing, so there are so many people to thank for their amazing work. But before I end, of course, wanted to thank Defeat Duchenne Canada for supporting our research. Without your continuous support, it would not be possible. Before I finish, so I do want to bring that up because I know that some people are interested. So genome editing is not science fiction anymore. There are actually people that have been treated, even in Duchenne. So both of those programs happen in China, but they were first patients were treated. And the safety profile, at least from what's being reported, seems to be okay. And I also am aware that US is spending a lot of money to trying to bring genome editing into a population of rare diseases. Right now, there's an ARPA-H call. So it's an advanced research agency programs for health, and they literally are going to spend tens of millions of dollars to bring genome editing into a clinic within 2 years. So I do imagine that in a matter of years, we are going to see more and more genome editing applications coming into clinic. Thank you.
Thank you very much. That was very informative and very exciting as well. Maybe we'll just move it out to the audience. Are there any questions from families? Or anyone, for that matter?
I had a question about the gene editing. So what you most talk about is making changes to the exon. Is there research looking at, or is it the same to make changes to the intron? Because that's where our sense mutation happens to be.
Hello? It is, yeah, for sure. It's actually much easier to make a change in the intronic locations because we are less worried about making a change in an exon inadvertently. So it's actually easier for us.
Oh, we have a question over there.
My question is about gene therapy. I know for like last 2 years, people talking about excluding exon, like first exons, including 8 to 11. So I want to know if there is any update regarding that. Thank you.
I think that's, so basically the reason for exclusion in those areas is because it has been seen that you have an immune reaction, that specific areas are highly reactive to actually have an immune reaction against that protein, that specific region. Like, we'll see down the road how everything goes. I think we need to develop, this is my complete personal opinion, okay? I think on the clinical trial settings for now it's going to be like that. I think down the road probably we need to develop better immunosuppression protocols to actually create that. The same thing, like the idea of the peanut butter, like just to expose it and get better over time, is basically how you increase what we call Treg cells. So for you to understand, like the Nobel Prizes that for medicine actually won about this immune reaction on Tregs, which is basically how you create tolerance to a foreign protein. I personally think it's going to change down the road. But in terms of getting approvals and all that stuff, I think that's the situation now, and we'll see.
I had similar question, I think, and maybe you've answered already by this question. But is that what I wrote is that you were talking about the immune reaction to protein. So that goes with the early exons. Is that the immune reaction to the protein or to the virus?
To the protein
To
itself.
the protein itself.
I mean, you can have an immune reaction to the virus, which is usually the adverse effects you will see really early because it's like a viral infection, okay?
Yeah.
It's like you fight against that. And then around 3 to 4 weeks after, you can have what we call adaptive immune response. For you, like it's basically you start looking at these proteins get expressed, and then you start attacking it. And usually, like in terms of the protein itself, since it's going to express mainly on the muscle, basically what you see is an inflammation on the muscle. It's what we call myositis or immunomediated myositis, IMM. Okay. And the risk on that is that you might actually worsen the disease.
Yeah.
Because you're actually producing more muscle breakdown and you're speeding out the process. So that's why I think we need to look into, again, there's in general in the clinical trials, what we do now is that we, like all the patients are on the Flazacort, then we put in higher levels of steroids, usually prednisone, and keep it like that. There are other trials that are actually using another immune suppression scheme, like sirolimus, tacrolimus, things like that, to actually target different areas on how do you immune suppress that. But we still don't know. I think my personal feeling, like having been working on this, is like we need to talk with the organ transplant people, because I see, for example, myositis against, like myositis in patients that have actually received an organ transplant. But organ transplant, basically, you're throwing the whole DNA of another person there. I mean, it's way higher riskier. And so I think the experience is there. We just need to grab what they have actually done, understand how it's the immune suppression protocols. If that helps.
Yeah, that's, thank you. So pretty much the protein reaction, the immune protein reaction is just to those certain exons.
For now, yes, but we don't know. I think we still need to learn a lot. I mean, I think there's a way that to predict which patients might or not go into that. For example, there's this complex of proteins that it's basically tells you how you might react against how you might present the protein. So basically, the cell actually will grab, digest the protein, and present in the surface of its muscle and say, I don't know what is this. That protein is called MHC, or in humans' bodies, HLA. It doesn't really matter, but it might be like really good at presenting that, or it might be that the binding is not really good. So you sort of can, I think you can actually predict it. For example, in organ transplant, they actually do this genotyping, that's genetic testing, to actually understand which, like if you are, how you're actually expressing, or how you're going to, or you might actually present an immune reaction to it. And I think we still need to look into that.
So you're pretty much like pre-testing the reaction, what it might be once.
Yes, I would love to start doing that. But I don't know how many people's actually doing it. I think we should start doing that. I think it's all like on the table to actually look.
But it sounds expensive.
Well, yeah. I mean, I think HLA genotyping. Yes, it might. I mean, it might. I don't know. Like, we'll see. We'll see how it goes. But any clinical trial is expensive, like super expensive. And developing these therapies are really expensive in general. So, I mean, I think it can be done.
I did have one more question when it came to the gene editing, because I'm just wondering if this method could be used for large deletions as well. Because the duplications, when you look at it, that sounds quite explanatory. But if you have large missing part of that chain, how would that work?
Great question. Yeah, duplication is a bit easier because we already have everything there. We just need to remove something that is extra. Deletions, we have to put some stuff back. And definitely it is possible. We just published a paper and probably came out 3 months ago. We can do it, it's just still not efficient. So, but what I can say is that even 2% editing efficiency in the heart, in mice at least, completely rescues cardiac physiology in dystrophic mice. So I know that it's doable, efficiency is not there.
Thank you.
If you ask me, I think we, like personally, completely personal opinion, we need to look into the vectors and actually increase the capacity of it. Because for genes like dystrophin that are so huge, like in terms of cure, like I'm talking about cure, not talking about changing a Duchenne to Becker. Like for me, it makes sense to actually go for looking how we can improve the vector component, so you have a larger capacity, you can put the whole gene there. Or eventually think about gene editing and how improve the efficiency for like the deletion component.
Question. So when there comes a time when gene therapy and gene editing are both available, what are some of the major factors we should consider? You know, I don't have very much knowledge like in this per se myself. What are the major factors we should consider when deciding between the treatments? Are there some points in the sickness where one will make more sense than the other? Or will both have similar kind of outcomes?
So can I take this one? Okay, and then I'll let you. So I think one of the things you need to think is like when you build a house, like you have a hammer, you have a saw, you have, I don't know, different tools. And that actually goes to what you were saying, like deletion, how does it work on gene editing? I think you have different tools, and depending on the change or the mutation you have, you might actually go into one direction or the other. And eventually, if you are lucky enough that you have all the tools there, then I will ask myself which one is the one that eventually has the best benefit with the less complications. That's the way I think, you know. Zhenya?
So one of the advantages of genome editing, and I'm saying like it will come in the future. So genome editing allows to fix on the level of DNA, and if we are able to hit stem cells of the muscle, satellite cells, it means that every progeny of that stem cell is going to be fully corrected. So that's when we are going to be talking about cure, not just treatment.
Is that clear? I think this is important to understand. Remember when I was telling you that dividing cells, And then if you actually put back the gene, but you have a dividing cell that dilutes over time because the piece of DNA that you're putting back is just floating there. It doesn't change the DNA. Gene editing does change it. So eventually, that actually makes even more sense. But if you have point mutation duplications, at least now it seems to be more feasible than actually putting back a whole deletion. Yeah?
All right. With respect to gene editing or gene therapies, both use currently AAV9 or AAV8 vectors. Has there been any advancement on the redosing? If you've been exposed to AAV9, is there work on exons or VLPs or anything like that going on?
Yeah, so that is a big issue right now because if the titer of antibodies is high, right now people are not doing it. I think here, even at SickKids, we are talking to immunologists, what would be the best way to actually do it? There are some people are attempting to do the treatment in patients with high IgGs, and they can do plasmapheresis, for example, or to start maybe to transiently eliminate B cells that produce antibodies. So people are working on it, and this problem must be solved because there are so many kids have been on microdystrophin, and if the second treatment is required, we will need to solve this issue for sure.
Yeah, there's also work on actually modifying the capsid. Capsid is basically the outside of the virus, so you can actually change it to eventually avoid those complications, like the antibodies. So for you to understand, it's like when you are exposed to AAV, you can't redose because it's like the vaccination. You actually put it exposed to the virus, then you develop these antibodies. So you will have neutralizing antibodies, and eventually that's an issue in terms of like reduced effect and eventually even also immune reaction.
But I guess another thing that probably is going to come, it is lipid nanoparticles or EVLPs. So when we don't need to use the virus anymore. It just right now, being on many grand panels, I can tell you it's not there yet. But people are working on it.
So with gene therapy, right? Like there are a lot of immunoreactions in the body, right? And gene therapy in itself is not very easy on the body. So when, as parents, we make a decision to go with gene versus exon skip, right? So what are the factors we need to think about if we are going with gene therapy? And then the second part of my question is with the recent news with the Sarepta gene therapy, right? Like, what are the learnings from those couple of incidences that has happened that the broader organization is kind of learning and trying to prevent that, because those are incidences which were, I believe, in non-ambulatory boys in general, but like that creates a scare among me as a parent whether I should pursue this gene therapy as an option.
Yeah, so that's a great question. So when you think about antisense oligotherapy versus, I guess, first of all, again, you need to look what's feasible or not, and then say, can you do gene transfer, or, for example, if you have a change in an area that you have a high immunogenicity, you will not be able to go to that. It's not, or eventually, if you have a mutation that is not available for antisense oligotherapy, I mean, there's not much options there. One of the questions you always need to ask for antisense oligotherapy is like, how good is the concentration in the muscle? How good is actually the expression of it, and how much is actually restoring? And if it's actually not only, and this is actually for both, that it's important you ask your physician, is like, how good is the restoration of the protein? Usually they show you the media, okay? You need to know the variability as well. How big is the variability between individuals? Because if you have a median of, I don't know, like just to put a number, like 10%, it might go have a 20, but it might be as low as 1%. So the question is, and we don't know, is where you're going to be located. In terms of the hold, like as you were talking about the hold on Roche, for the therapies, I think the data, this is my complete personal opinion again, the data for older patients wasn't really strong there. Actually, there was a good editorial from Mariam Scooey, Jim Dowling, and Laurence Servant about that, discussing about the expression was actually great. It doesn't mean that from the functional component it might actually working. So that's what they were saying. I think the older patients have a way higher risk, and the benefits is not so clear. And if you ask me, probably, I think the medications works, but the younger the patient, the better the outcome. And this is actually also driving to discussing newborn screening for Duchenne patients. And this is a current discussion in Ontario, or if you didn't know that. And preparing for how we actually develop these kind of things. Because in terms of complications, the babies, they tend to tolerate basically everything. Like, it's way safer to do gene transfer therapy in a baby, newborn, than in 18 years old. Way safer. It's lower the dose, the immune reaction is way lower, and they have this tolerance to foreign proteins that, I mean, I've been done a lot of gene replacement therapy in the SMA population, and I think it's safer than in the older patients. I don't know if I respond your question.
That's
yeah?
fair.
I think I started to
Yeah, because currently we are exploring my kid is now almost 4, so we are exploring all these options, right? And we looked at a couple of options, but all these questions and everything arise, right? And it creates a doubt, right? Because all these, when immunosuppression reactions happen, right? So do you want your kid to go through that? Are you introducing something in the body to correct one thing? You're introducing something else, which might in the future, when you look at that brochure, hey, it can cause this, right? And those are a bunch of terms that you don't want to, read at, right? And it creates a fear whether, you are taking the right choice, or you are just treating your kid as a guinea pig to go through that, right? Like it's a really difficult choice from that perspective, right?
Well, you're completely right. It's completely individual decision. I talk to a lot of parents, and some parents are going to sign me to the trial to do gene editing tomorrow if it's possible, and some people, I'm not going to do it. So it's completely individual choice, and every family is going to make this decision. But maybe to answer your question, so people with those adverse effects still happened with older generation of AAVs, I would say. The new generation myotropic, when we are going to reduce the dose, I think they are going to be safer.
Okay. Yeah. So it's just dose dependent, like the more the dose, the more adverse reactions, and with the newer virus,
No, not only that. I think the dose is really important in terms of the immediate reaction against the virus, because most of the death that has happened were related to a liver insufficiency, because the liver gets the biggest hit. Remember, we were talking about the promoter, you don't want to get the liver, but the truth is the AAV9, AAV8, or even the modified ones will hit the liver first. So you want to reduce it as much as possible, because although you actually have, that's why you want to have a more tropism for the muscle and try to escape the liver as much as possible. Now, even if you, I know it's not the same, but if you read the Tylenol, what you can get, that's scary as well. But I'm not saying it's like gene transfer therapy. It's like, no, you need to take it serious. I think you need to think about this as organ transplant. And what you actually mentioned, guinea pig. Well, I never had my kid in a clinical trial study, so that I can't say that. But I think we need to reduce the idea of this idea of guinea pig component. But I know you guys feel like that. It's like, I understand that. The thing is, I think you need to partner with whoever you're going to work with. You need to feel comfortable. You need to feel that that team has your back. You need to feel that if anything's going to happen, they will be there with you. It's basically going to be your best friends for the next 2 or 3 years or even more. So I think you need to go to a center that you really want to work with.
Yeah, I guess my use of the word might not be correct over there, but yeah, my feeling for that was something else. Okay, and my second part of the question, we talked about the genome editing, right? And then there are some therapy comings up which are individual based, right? In like there's coming. Do you think that in DMD will come up with therapies which would be individual based, or would be more broader, just the exon based or skip based therapies?
Yeah, in my lab, we do develop a lot of individualized therapies. So it's a very specific mutation, and we are trying to find a way how to correct that specific mutation. So it's definitely going to be available, but it is not a generic therapy. So who is going to be paying for it and how it's going to be reimbursed and so on, those are still big questions.
Actually, that's a big one. How you actually move from the lab to actually dosing a patient. In Canada, those pieces are not really well established. We have been working on this with like a lot of people. CIHR is working on that. It's way more established in US than here. But how we actually do it, it's not really well established. Okay.
So
But we're working on it.
You're working on it. We're gonna, and we do have to move on, I'm very sorry, but I do believe that you'll both be around for a little bit longer, so if anyone has any questions, you're welcome to grab them and continue the conversation, but we do need to move on. So thank you so much, I really appreciate it.